Thank you, Victoria. And thank you, Priyanka, and the organizers for the invitation. So yes, I am Francisco from Genomics England. And today I will give you a brief tour through some of my research in the area of explainable, trustworthy AI. I will start by introducing two key dimensions of trustworthy AI: explainability and transparency. Then I will illustrate this with two application cases, one of them using multiple types of biological data and network approaches to integrate them and make predictions of drug response. And the second application case will be the use of generative AI to support the curation of biological knowledge using large collections of scientific papers. And I will conclude with some takeaways and recommendations. So explainability and transparency are two complementary dimensions in enabling trustworthy AI. On the one hand, you have explainability that can be defined in different ways or approached in different ways. For example, you have model interpretability, which is about the understanding the decisions, the outputs of an AI system. We also have research in the estimation of feature importance that are relevant for predicting and validating key biological indicators that influence an AI prediction. You have other sub-criteria within this dimension. And then you have the dimension of transparency, which can be achieved through data transparency and quality. This means, in many ways, providing clear documentation of data sources, your data collection methods, your quality assessment techniques. You have also methodological openness that can be achieved by providing detailed descriptions of your models, including the model architecture, but also the development process and the types and characteristics of data used to train those models. You have other key aspects such as resource reproducibility, and of course, the ethical and stakeholder engagement that is required to increase or gain trust in these models. These two dimensions can operate independently, but at the same time, they complement each other because they can support, enable, or inform the other dimension. And these are the key topics of my presentation. I will next illustrate these two dimensions and how to achieve them using two examples. The first one is the application of network approaches for multiomics data integration. In this example, you will see not only how different types of omics data can be integrated to make predictions with potential clinical relevance, but it addresses transparency by providing clear data integration methods and explainability by showing the complex biological interactions underlying those predictions. And the second use case involves the application of Gen AI to support biological evidence annotation that is important for supporting our updating, curation of our diagnostic panels at Genomics England. In this case, you will see that this approach enhances transparency by providing traceable sources of evidence and improve explainability by generating human-readable summaries of complex evidence that can be used by the human expert to make a decision on whether to update the evidence connecting a particular gene variant with a particular medical condition. So the first example, multiomics networks for differential drug response prediction. In this case, the goal is to integrate different types of omics data obtained from patients exhibiting different phenotypes. For example, types of disease, subtypes of disease, or different manifestations of a phenotype. So you may have a patient group A versus a patient group B. That could involve different phenotypes, also different clinical responses or responses to treatment. And then the idea is to generate these different networks consisting of different types of data. So you have different layers of omic information for each patient group. And next, the goal is to integrate these these layers of omic information coming from the two or more patient subgroups into a integrated differential network. And then with this differential network, we can make predictions, differential predictions on this network. And this could be, for example, predicting the response to a particular drug. And then by doing this, we can use the network-based representations to explain how this prediction was made and the relevance of the data used for the prediction. And this is something that we develop and share within a single package, our package called Dr. Diamond. And this is a collaboration with colleagues in Germany and Singapore. So among the use cases that we investigated, we asked the question, can we use this differential network approach to estimate drug responses in cancer, in particular breast cancer, considering two subtypes of cancer that are clinically relevant, ER-negative patients and ER-positive patients. These are clinically relevant groups because they have different prognostic and drug response characteristics. So we use data coming from the TCGA, including the different transcriptomic, proteomic, and phosphoproteomic data layers. Also, we investigated the value of integrating metabolomics data. Then, in this case, for each cancer subtype, we built single-source omic networks that are here illustrated with different colors. Then, for each condition, we establish interlayer associations, that is, connections between the different omic data layers. Once this is done, we estimate a differential network, which is basically a reflection of the differences between the two patient groups. So once we have this, we can query the tool with a drug or a collection of compounds, and then considering the targets, the known targets for these drugs, we can make estimations of drug response, we can rank the drugs, and then we can investigate the underlying interactions for these predictions. So one key observation is that our predictions of drug response correlate with the known responses to these drugs using in vitro models. We use in vitro drug response data as a surrogate of clinical response, as an approximation, and we found that correlation. On the left side of this picture, you have just a plot summarizing the ground truth for our analysis, coming for the Cancer Therapeutics Response Portal. So here, basically, you have a comparison of patient groups, a comparison of the cancer type groups, ER positive versus ER negative, observed in the cell line data. So as you can see, you have different levels of response defined or estimated using the full change in drug sensitivity. And then using this gold standard, we compare our predictions using the multiomics network approach. And apart from estimating the correlation between those predicted and observed responses, we can analyze the classification power using, for example, AUC ROC curves. And as you can see, there is we show the potential of this approach to make good prediction, especially when we focus on the top rank drugs for this data set. So apart from showing the potential prediction power of the method, we also illustrate how a network-based approach can be used to support the explainability of these predictions. So, for example, giving a drug like momelotinib, then we predict a drug response score, 0.63, which is relatively high in comparison to the other observed predictions. Then, after that, we trace the targets for this drug. In this case, you have JAK1 and JAK2. And then using this information, we can look at the differential ages for these targets in the differential network. So as you can see here on the left, for JAK2, there is no differential, a differential integrated interaction score, which is a reflection of how that drug is predicted to affect the reference patient group. But in the case of JAK1, you can see that for different interactions connected to the target, you have a stronger differential integrated interaction score. And that integrated interaction score is a reflection of the estimated strength of the interaction between the drug and these targets and these components in the reference network. So apart from that, we can continue going back into the network representation and say, okay, now we know that this drug is highly differentially, the network is highly differentially responsive to this drug. Can we go to particular patient groups and estimate the importance of the given targets for this prediction? And we can see here that in the case of JAK-K1, you have clearly stronger interactions in ER minus, negative, than in ER positive. So these plots can be used to interpret the predictions for a given drug, considering known targets. And this is consistent with evidence observed in the literature. And we have other application cases. And just the second part of my presentation is the use of generative AI for supporting diagnostic knowledge curation. The challenge here is to support our diagnostic pipelines to go from millions of variants observed in a patient's genome for a given condition, how we go from this amount of potential variants to fewer than 10 variants that can be used for supporting a diagnosis. So to support that, we have a knowledge base called PanelApp that maps gene to diseases using evidence coming from different resources, in particular the scientific literature. The challenge of updating these panels with the latest, most accurate evidence is high, is time-consuming, is complex. So the challenge of, for example, for a given panel, for example, a panel describing associations between a disability, we have this evidence, supporting evidence criteria, green, amber, and red, with green genes representing those associations that are ready to be used in the clinic. So the challenge here is how we continue updating these panels using the scientific literature. So to support this task, we define a workflow that is powered by GenAI, and the objective is to augment the curator's ability to extract, not only to identify associations, gene disease associations in the literature, but more importantly, to extract evidence supporting those associations so that the biocurator can update our panel app database. So here you have a description of the workflow that begins with searching for genes in the literature, then encoding those articles and sections of these articles in a format that is readable and useful for machine learning. Then next, we retrieve the articles that match a particular gene disease query. If the text is found in any of these articles, then we use that information as context to query a large language model to find associations and the supporting evidence. Then the outputs are provided in a user-friendly structure format to allow expert assessment and decision making. So I don't expect you to read this slide, but this is basically to highlight that the inputs, the query inputs given to the large language model includes a number of aspects and information that are relevant to obtain accurate and reliable associations and supporting evidence. We also ensure that these prompts induce the outputs that are also well structured and readable. And here's an example of the outputs where you have not only the prediction or the estimation of an association between a variant and a condition, in this case, intellectual disability, but also you have an enumeration of the evidence supporting that, and that evidence is structured into different levels of evidence. For example, evidence related to observations found in patients, observation related to different mechanisms, observation supported by functional studies in animals. And then, in the end, we provide a brief summary of the association and the supporting evidence. And this organization allows a fast and accurate assessment that is conducted by the biocurator. And here you have just a brief illustration of the kind of results that we have obtained so far, in which we have, in the case of the mental disability panel, we have been able to verify existing associations, but also upgrade or promote new associations that are suitable for clinical care. And beyond this pilot study that was done in cooperation with AWS, we are expanding not only the data scope of the system, but also we are implementing automated, user-friendly pipelines to allow the users to have these outputs on a regular basis that can be assessed in an efficient way. So to conclude, my takeaways are that explainability and transparency are multifaceted and synergistic approaches. These two examples illustrate that interplay. For example, we can see that they mutually reinforce aspects in a given application that, in the end, enhance AI trustworthiness. This is observed also in the case of network approaches, where by revealing complex interactions we boost not only explainability, but also transparency. In the case of the biocuration project, by doing this, not only we're accelerating the annotation process, but also improving the generation of human-readable summaries of evidence that are also useful for accelerating the biocuration process. And I thank my team at Genomics England and our partners at AWS, and also my collaborators in Berlin for this work. Thank you for listening.
Thank you very much. Do we have any questions in the room? So we have one here from John.
Multiomics network. Have you tried other data types going into the AUC? Methylation, performance structure, knowledge, and so on?
No methylation, but that's the approach is can be adapted to include other layers, definitely. But the methylation part, no, we haven't investigated that yet. In some cancer types, I would agree that this could actually enhance the prediction performance, in particular in cases where we know that methylation is a prognostic factor. But in the case of breast cancer, we didn't investigate that dimension.
Okay, I think we had a question in the row behind.
Hiya, thank you for your talk. I'm right here. Very quick and boring question. Which LLM embedding model did you use for the RAG pipeline, and did it affect your results very much? Because there are specific ones that are trained on or fine-tuned on PubMed, and there are more sort of normal ones. Is normal semantics enough to extract the summaries from each paper or not? Thank you.
Yeah. So the embedding model that we're using, so we are using Claude model, first of all, within the Bedrock platform of AWS. So we are using the face model for indexing. And then we have, for the embedding, actually, we try different approaches. If I recall properly, the optimal approach is the one provided by AWS Titan, I believe it's called. So it's the AWS embedding model, and that's so far the optimal approach, and also the most efficient. Does it answer your question?
Yeah, thank you.
Okay, and we'll take one more question just over there from David.
Hi, thank you. And again, related to the RAG at the end, I was interested, you had Claude was returning a summary and then, which I think you described it as like a chain of reasoning. Was that actually chain of reasoning? And how much do your curators actually, do they spot problems in that chain? I'm curious how often that happens, because you talked about trying to encouraging it to give something so it's quite conservative. Also, on a separate angle, I'm going to squeeze in two, sorry, Victoria. How much do we, Tim Hubbard talked about the problem of reproducibility of research. Is there any chance that in using the LLM, you're amplifying existing fashion in science, and there's a bias there?
Yes, thank you. So the first question, the prompt that you saw there is just the output of a journey of prompt engineering, where the biocurators play a key role in providing feedback. So you mentioned a couple of points related to not only accuracy, but also the reliability of the outputs, including the consistency. So all these elements were used by the biocurator to help us fine-tune or engineer the prompts. So definitely that was part of an iterative process until we found a prompt that was not only structured enough to produce structured outputs, but also reliable enough and consistent enough. And the second question, I think that's a crucial question. In general, not only in the context of a literature review, I think it's one of the challenges for AI, how you reinforce existing modes of thinking and that rely on existing probably biased approaches to research. My hope is that by allowing the users to make the final decision and providing the best available evidence to support that decision, we can reduce some of those biases. Of course, that's not a guarantee, but I think it's better than just providing an output and saying, this is your prediction, make a decision, yes or no. So that's my current approach. I hope that's big. Okay, thank you.
Okay, please join me in thanking Francisco.
Thank you
again.
very much.